speed up a little bit. What I wanted to cover was clinical trial of new treatment for Wolfram, what we're trying to do to contribute to that in Birmingham with Dewey, Susie and others. A request for help with blood samples from some of you, if it's not been involved, and then if I can finish in time, an update on the European registry for Wolfram with Caitlin, who's here as well. And as I go, and we've done the update on stem cell research in Oxford with Pinnacle, which we've done first. And as I go through, I'll introduce the different researchers that have done the work that I'm going to talk about as we go. So many of them are here. So my version of Fumi's slide is a timeline to how we go about from a condition to treatment. And what I wanted to say is, obviously the first thing is trying to find the cause of a condition. And Fumi has led the way, a long way, in going to try and understand the cause of Wolfram, but we don't know everything about it yet. We're still working, there's still gaps in the knowledge. Once you found the cause, then got to try and find a drug target. Got to find something that medicines might work against. And Fumi's mentioned the drug target that he's talked about, and I'm going to mention another drug target that we're looking at. Once you've got a drug target, you need to do studies in cell models to see whether it's going to work out all right. And if it's a new medicine, you probably need to do trials in mice or animal studies as well. Once that's done, you can then do studies in humans to find the right dose, find out whether it's safe and whether it's effective in people, and that's called early phase studies. And once you've done early phase studies, you can do what you've probably heard of, which is a randomized controlled trial of a medicine with people, where you would give half the people the study medicine and half the people a dummy medicine. And you follow everybody up for a period of time, and at the end of it, you what's called unblind it, and see whether the people on the study medicine have improved compared to those on the dummy. And if you can do that and demonstrate it works, you can then get marketing authorization, and you can put it in the formulary for a recommended treatment for a condition to go. And I just say it just because that's for any condition, so that you can understand that that's the process that you have to go through. And along the side of it, you have to find something to measure to show whether the treatment works, and that was the biomarker that Fumi talked about. And then once you've found the biomarker, you've got to prove it's going to measure what you think it's going to measure. That's called validation. And then it's no good having a blood test that tells you in the test tube that something's working. You've got to find an outcome that's relevant for people as well. What's called a patient-related outcome. I.e., people having the medicine have got to feel that they're getting better, not just on the blood test. So either the eyesight's got to get better, or the balance, or something like that. So there's quite a process. People say that if you're going for a completely new molecule, it might take about 15 years from the start to the end. If you can use a medicine that's already used for something else and repurpose it, use it again, you can probably knock about 5 years off that, and then maybe you're looking at about 10 years or so by that sort of time. And what I want to do is to go through where we've got to on this pathway and show you hopefully how things can be speeded up a bit. So the goal, as Fumi has said, is obviously we want to find a cure for Wolfram, reverse the disease process, maybe restore useful vision, and Fumi's outlined really nicely how people are going about to try and do that. And there are some researchers in Montpellier, in France, who have got some very exciting research that's going on, looking at potential gene therapy and stem cell therapy for eye disease and that. And Guy Liniers came and talked last year, and maybe we should, with Virginie's help, perhaps invite a French researcher next year to update the work that the Montpellier group are doing, because it's really, really impressive. And they're working on a mouse model, kindly provided, I think, by Sulev Cox in Estonia, and also before that from Fumi. So Virginie, can you stand up, Virginie, so that we can say who you are? That's the thing. Virginie Picard is here, a scientist and a representative of the French Wolfram Association, who supported the research here. So Virginie, thank you for coming, and thank you very much. Okay. Sorry, I'm going to pick on people as we go around and introduce things. So, what we've been doing in Birmingham, we've been trying to find a target, and the target has been identified here by my colleague, Dr. Georgia Nagy, who is a scientist who's worked on other diseases, things like Alzheimer's disease. But what seems to be helpful there was good for us. And she shows a cartoon of a cell cycle, that every cell in the body goes around a cycle of G1 phase called growing, And then once the cell's grown a bit, it has a stationary phase or a settle phase, then it grows a bit more, and then it divides. And many cells in the body go around that cycle. But to make sure that it goes on without the cell getting ill or getting problems, there's different checkpoints. And there's a checkpoint at the first growing phase to say, is the cell healthy? Has it got the right proteins? Is the DNA not damaged? And then it's allowed to go through the next bit of checkpoint. And then there's other checkpoints there as well. What Serge found was a protein that controls this checkpoint here and stops cells going on. And this protein, called P21, when it's present in cells, seems to control going through that checkpoint. And that's important because if a cell's not healthy at this point, because the DNA is damaged or it's got stress, like in some Wolfram cells, it goes through that checkpoint and off into cell death. And this P21 protein seems to stop it going down to the cell death pathway. So by blocking it there, it keeps the cells in that first stage and gives the cells a bit longer to get healthier and to clear the problem protein. So that was the target that Serge suggested. And this was the work that was published by Dr. Zatika a couple of years ago, showing that this was the case. Malgosia, can you stand up at the back so we can say who it is? Dr. Zatika is our senior scientist here and did this work. And what she did, apart from the fact that my slide has completely gone wrong, all the things I was going to show have disappeared off the end of my slide here, was that she's looked at cells in controls, and we took human brain cells from children who had a form of brain cancer called neuroblastoma, and we grew them in the lab because you can't do a biopsy of people's brains, you can use brain tumor cells. We took out the Wolfram protein out of them, and then we measured the amount of the protein P21 in it, and we found cells that didn't have this target seemed to have lots of cell death, but cells that kept that target seemed to be protected. So that was what gave the idea, suggesting that this protein might be helpful to keep cells alive. And I really hope all my other slides haven't gone wrong as well. I'll find that now. So Dr. Dewey Astuti did what's called a high-content drug screening of a small drug screen, looking at drugs that are already in the formulary that we know can increase P21 protein expression. So there are quite a lot of drugs already, and drugs that are already licensed for use in children and licensed for use in adults. Dewey, can you stand up please at the back? Dewey Astuti is our other postdoc and worked very hard in Dr. Naji's lab to do this. So she screened 28 drugs from a very industrial-like process to do so. Again, using these children's brain cells from brain tumours in children where we took the protein out. And she found 5 medicines, five drugs here, which seem to work in these cells with Wolfram syndrome in our model. And as I said, these are drugs that are in the children's formulary. They're already being used, but for different conditions. She selected the best one to take forward and chosen one of these. Now this one works. And then what she did in the high contact screening was use this study drug in different concentrations, each one 10 times more than the other, and looked to see whether or not, compared to control cells, whether or not the Wolfram model cells showed more P21 protein in it. And to describe what she could see is that different drug concentrations could raise the concentration of our protective P21 CIT by about between 10 and 15 and 20% on those. So it looked promising that this drug was doing what it wanted us to do, what we wanted it to do. And then she wanted to measure whether, by giving the medicine, the drug, it was protecting cells from cell death. And she looked at the proportion of cells that were positive compared to those that were negative, at the different drugs, and could show that when the drug was applied, only between about 3 and 5% of the cells were dying compared to the bigger numbers before. So it looked like that this medicine is keeping cells healthy and stopping them from dying. And then thought that was okay for high content screening, then wanted to go on and then have a look in cells individually, in tissue culture in the lab on a smaller scale. And Dewey again has done this. She's looked at control cells and cells with our 2 models of the disease, KD2 and KD3, and she's given those different control cells different concentrations of the study medicine that we want, and measured the protein expression, and she could show really nicely that the expression of our special protein, P21, goes up by between 20 and 40% in our 2 cell models, looking at this. So that looked good. So fingers crossed that was doing the job. Was it protecting the cells from dying? So this is work, first of all, done by Dr. Renica Diaz. Renica, are you still here? Renica, stand up. Dr. Renica at the back there. Stand up again, it was too quick. So just to say that she did this. So this is looking at protein at the RNA level. And first of all, she looked in control cells, first of all, to see the effect of the drug and the expression of a marker of cell health. And this is a marker to say that cells are stressed, and she could show really nicely that the cells get less stressed in the controls, but most importantly, they get less stressed in the Wolfram models as well, showing the cells look like they were getting healthier. And in fact, it worked really nicely in both Wolfram cell models. So thought that was looking promising. And then Dewey went on and did some work again looking at the protein level at a marker of cell death called caspase, which Professor Urano mentioned on one of his slides, and looking to see if this marker of cell death went down. And we were really pleased to see that in the control cells with the study medicine, less cells died, but most importantly in the Wolfram cells with the study medicine, the rate of cell death dropped as well by between about 20 and 30%, something like that. So this was looking really promising that this particular medicine that's used for other things looks like it might have an effect in the lab for Wolfram syndrome, which is obviously what we want at that level. So, going back to the timeline for treatment, we know a lot of the cause of Wolfram syndrome with the work that Foodie's done and others have done around, although not complete. We've got another drug target, P21 protein here. We've got studies in cells, cell lines we've had, which seem to show it work. Just like Fumi's medicine and our one, probably don't need to do trials in mice because it's already licensed for use in people for different conditions. So we know that it's not going to cause cancer or long-term things like that. So it cuts this timeline down by at least 5 years. It makes the whole process quicker. What we've got to do next is to do studies in people, to find the right dose to use, and to prove that it's effective in people, just like Fumi was describing as well, and then on to randomized trials. But the other crucial bit is we need to know that there's a biomarker, something we can measure in a blood test to show that this medicine is working in people. Because you need both bits. You need not just the medicine, but a way of proving it or showing it's working. So where next? We've applied to the equivalent of the FDA, which somebody asked that question about Federal Drug Administration approved here. The European version is the European Medicines Agency. So we've applied to the European Medicines Agency to designate this study medicine an orphan drug with the indication of the use for Wolfram syndrome. And the application went in September, and we hope to hear in February. And the importance of that is it shows that other people in Europe think that there is some evidence that this is going to work, and that we will then be able to conduct a clinical trial of this medicine in people with Wolfram. We have to do that stage first. We've also applied to the European Union, to their grant bodies, for money to do an early phase clinical trial. The outline has gone in. We hope to hear about that before Christmas, and then a full application has to go in, and then if we got that far, we would hope to hear about it next autumn or something like that. But at the same time, we need to see whether our biomarker works. And what we're doing is testing in the white blood cells from affected people, their mums and dads, and healthy volunteers. And what we're trying to do, effectively, is from a teaspoonful of blood sample, take out the white cells, measure the amount of this protein, P21 in it, how much protein there is in the cells, and also how much stress there is and how many of them are healthy or not. Then we take those white cells in the lab with Dewey, and we treat them with the medicine that we've got, and then we look at those and see whether the P21 protein is going up as we think it is, and the stress levels are going down. So really important to move from the lab to people's cells before you actually give the medicine to people themselves. And it's a nice way of looking at people's cells without giving them the whole medicine. And it's also important to see whether people respond differently. And it may be, as somebody asked the question earlier, whether some people with different symptoms respond differently to other people. And this first test will help target who needs to go for the treatment, or to have this treatment. So, What I'm asking for this here, which I'm mentioning, is if you're affected by Wolfram or you're a parent, or if you're a healthy volunteer that has nothing to do with Wolfram, and you're interested, we'd be very, very like to hear from you really, to sign a consent form and then ask, if you've not done it already, and some people have, to donate 2 teaspoonfuls of blood. And then this information on the biomarker will help us with the clinical trial application and actually show that we've got a marker to go if we get through to the next stage of the European one. But it's also really important that you don't have to take part if you don't want to. Some people have already taken part and very kindly gave us blood last time, which I'm very grateful for. The blood test itself doesn't actually help you directly, but it does help us in the process of developing a biomarker for the future. So that's where it is. If you're interested, then I'll be around over lunch in the side room that says Dr. Barrett, and I can do the consent form, and we can do the blood test here, and then Dewey is going to hot-foot it by car with a box of the samples back to the lab, and then spin them down and freeze them. And I think with Dr. Naji, we'll be coming in tomorrow to process those to get that done. So that's if that's something that you've not done and you'd like to do, you can. But as I said, we have got samples already, and we don't need everybody to say yes. So don't feel that you've obliged to. That's also really important. So that's what I was going to say. Finally, I wanted to say a big thank you to the French Wolfram Association, Association Syndrome de Francais, because the bit about the drug treatment was funded by them primarily, and it's been so incredibly helpful and exciting to have that grant to be able to do a drug treatment. And because of that, we may well be ending up with 2 medicines for Wolfram and 2 clinical trials. And I think 2 is better than 1 because it increases the chances of finding something that's going to work. And a big thank you to the UK Wolfram Syndrome, because they very kindly fundraised for us and helped us with some equipment in the labs to help us to do this work. It's been brilliant. And then a big thank you to Well Child, who've been a real supporter of Wolfram Syndrome going back probably 15 years, and it's been absolutely brilliant. Well Child have been supporting Wolfram Syndrome since about 1998, and if Linda Partridge was here, I would give her a big hug and say thank you very much to her, because it's been a great support. And then I mentioned our senior scientists who do all this work, Dr. Mogos Zatika, Dr. Dewey SDT, and then recently Renika Diaz has been helping as well. Joji Nalji, who's had this great idea of looking at this candidate in the lab, and Caitlin, who I'm going to invite to talk next. And then Dr. Sally Garani and Amy Arnold, who were working in the lab previously, and we've already talked about this, some of these colleges. So I'll stop at that, and then I'm going to hand over to Caitlin.